يعطيكم العافية. المحاضرة المحاضرتين القادمات we will cover the topic of sterilization and disinfection. So we covered growth and death of bacteria. We covered bacterial genetics and how they communicate with each other and how they grow and how they can incur mutations. And all this in the effort اللي هو المحاضرات القادمة the 5 lectures coming اللي هم عن sterilization, disinfection and antimicrobials. I'm studying growth and death. I'm studying DNA replication and mutation and all of that because I want to be able to control the growth of bacteria. I want to be able to limit the growth of bacteria, and I want to, if I can halt the growth of bacteria, and if I can achieve النيش اللي أنا بستخدمها مثلا to have zero cells of zero bacterial cells. So that's what we're aiming to do. We're aiming to get rid of our bodies, our surfaces, our tools of these agents. So how do we go on about this? So let's talk about disinfection, antisepsis, and sterilization. So sterilization, now let's go through some definitions. Sterilization is the first definition. It is defined as the process used to render a surface or a product free from viable organisms. So حكينا germination will produce a viable cell. So this will include bacterial spores, because the germination will produce a viable cell. The cell that can replicate زي ما حكينا, it is still a viable organism. So you are trying to completely remove all cells from all viable organisms from a surface or a product. That is sterilization. هو التعقيم. So another definition هو inactivation of all self-propagating biological entities, bacteria, viruses, prions, and تعريف آخر هو removal of all microorganisms including spores which are highly resistant. So كلهم يعني احنا بنحكي تقريبا نفس الشيء. In practice يعني بنحكي الواقع واللي بصير عندنا, we can achieve sterilization. Sterilization is really achieved by many equipment رح نحكي عنهم كلهم, but the most common equipment that is used by far is autoclave. Autoclave is designed to kill all organisms. The hardest structure of organisms is spores. If you kill spores, you kill everything else. لأنه is the most resistant form. So if you kill spores, you have killed all viruses, you have killed all bacteria, you have killed all yeasts and fungi and whatever. So what is the three? It consists of three things really. It consists of heat, pressure, and time. الpressure it's also إذا بدك تقدر تحكي كمان رقم 4 اللي هو steam, because steam itself can permeate, يعني بيقدر يدخل لأنه أنت بتستخدم مية بصير steam بدخل can permeate through structures and carry your temperature for you as well. So it is hot boiling steam water that you cannot elevate above 100 إلا إذا تحط ضغط. So it's 121 steam pressure for about 20 minutes, and this can destroy all bacteria. This can also يعني ما تقدر تستخدمه على كل شيء. رح نحكي إنه how we can use other things to sterilize sensitive matter, but that's basically the most important thing. رح نحكي عنهم كلهم هذول. Disinfection. What is the definition of disinfection? Disinfection is defined as the use of biocides. هلا نحكي شو يعني biocide, which aims to reduce the number of viable organisms. يعني أنت disinfection والantisepsis you are controlling, you're not eliminating. So you are controlling the number. مثلا أهم شيء زي مثلا لما تيجي بدك تأخذ تسحب دم من حدا أو تعطيه دم, بتجيب هالقطعة الألكوهول على قطنة وبتمسح المكان الجلد. أنت هون بهالألكوهول أنت you are killing a lot of the cells, يعني بتقتل منهم 70% 80% 90%. So you're reducing the burden of the bioburden. أنا بسميه بالbio burden. Bio burden is what will be what your body will have to deal with. So how much يعني إذا ما أنت ما نظفت, كان في مثلا 100 خلية على الجلد وعملت أنت حطيت النيدل, بدخل 100 خلية كلهم. So there's more burden on your system. اذا انت رحت نظفت قتلت منهم 90% راح ضل 10 خلايا، روح حطيت انت النيدل so you are introducing 10 instead of 100، معناته انت نقصت البيردن by 90% مثلا. So that's disinfection. That's basically what disinfection is. You are aiming to reduce the number of viable microorganisms. So on or in a product or surface to a level previously specified. This معناته انت sometimes you need to require to study each surface. What is the appropriate level to handle each surface? So they have to have the ability to inactivate the organism اللي هي انت إذا بدك تستخدمه. So they usually do you think they will be sporicidal؟ هل راح يدمر السبورز؟ Obviously not. يعني لأنك أنت هذا راح يكون شيء أقل من complete destruction of all living organisms، معناته راح تكون this is something weaker, so it will not be sporicidal. There is no disinfection mechanism other than that is sporicidal, but they can be sporostatic. شو يعني sporostatic؟ Sporostatic they maintain the environment negative that spores will not vegetate. So they are sporostatic, but they do not destroy the spores themselves. So they inhibit the germination of spores. هلا في disinfection و antisepsis نفس المعنى but antisepsis يستخدم is disinfection for living tissues, so it's the same meaning, but disinfection is used for surfaces and non-living organisms, whereas antisepsis is used for disinfecting living tissues. So this is basically you're trying when you're disinfecting a living tissue that you are performing antisepsis. Disinfection by definition is to be able to destroy many but not all microorganisms, and does not equal sterilization. لازم هاي نعرفها طبعا. Not all disinfectants are harmful to tissues. So ليش أنا ما بستخدم this? Because why am I doing this this distinction between living and non-living? Because disinfection, some things are corrosive like phenols مثلا and chemicals. I cannot use them on living tissues, so I have to have things that do not damage the living tissues to use them as a disinfectant for living tissues. That's why I give them a separate definition and I call them antiseptics. So disinfectant can be used on living tissues are called antiseptics. A biocide basically is a material, chemical or physical that has a broad spectrum that can destroy, inactivate many organisms of population over time. مثلا chemical biocides زي مثلا hydrogen peroxide, alcohol, bleach, cyclohexamide, phenols. Physical مثلا heat, radiation, pH. These are things that you can do. These are all biocides. These are the agents that you are using to disinfect or sterilize. هلا في زي ما حكينا sporocidal, we have the term bacteriostatic and bactericidal. راح نستخدم هذا الterm in antimicrobials and we will use them in sterilization and disinfection. Bacteriostatic means it's a term referring to the property by which a biocide is able to inhibit. So I am not destroying, I am inhibiting, I'm stopping growth. So that means if I lift whatever bacteriostatic agent I used, bacteria will continue to grow. معناته I did not remove their ability to replicate, I stopped their ability to replicate. I just stopped them from replicating and did not take away their ability to replicate. So you can see sporostatic, fungostatic, bacteriostatic. طب شو هالأهمية؟ يعني شو استفدت أنا إذا وقفته ما قتلت ما زحت عنه ability to replicate. That gives the body's defenses chance to overcome. لأنه مثلا عندك بكتيريا موجودة بالجسم it is replicating. خلينا نحكي arbitrarily مثلا مليون خلية كل ساعة، مليون خلية كل ساعة، مليون خلية كل ساعة. جسمك is not able to handle مليون خلية كل ساعة، مليون خلية كل ساعة، مليون خلية كل ساعة. So it is overwhelming your system. إذا أنا بعطي إشي bacteriostatic وقفت الريبليكيشن، فكانوا مثلا وقفوا عند 10 مليون ضلوا 10 مليون. So I'm giving your body time to defend and destroy these 10 million واحدة على واحدة until it catches up and kills all of them. So it is using your own defenses and giving time for your own defenses to catch up and destroy the microorganisms. So what is the sheep? اللي هو population growth control. So you're controlling the growth and you do not with the bacteriostatic agent reduce the numbers, but you are giving the immune system time to catch up. So the population control might just be enough for the host defense to control the remaining population. فهون هو bacteriostatic event. It is growing بتروح تدخلها انت على stationary phase right away. So this is the bacteriostatic event. Bactericidal بالعكس, a specific term referring to the property which a biocide is able to kill bacteria. هون there's no growth control. This is removal of the ability to replicate. So you are destroying the cell. It's irreversible. Bactericidal is irreversible, whereas bacteriostatic is reversible. So the killed organism can no longer reproduce when you lift the agent. So this causes مثلا lysis, irreparable damage to the DNA, مثلا irreparable damage to the proteins, which will kill the cell, and the cell is no longer able to replicate. So الأهم difference اللي هو the removal of the bactericidal agent does not cause the organism to regain its reproducibility. So here. The rate of killing of organisms حكينا this is the same thing. I don't want you to memorize any any معادلات, but I want you to know the concept of it. حكينا هاي قبل هيك إنه as you increase the concentration of the agent or you increase the length of time that the agent is applied, you are increasing the rate of killing of organisms. فحكينا هالمثال قبل هيك إنه مثلا حطيت أنت concentration of alcohol, خلينا نحكي مثلا خلينا نحكي concentration X مثلا. concentration X فياته 100 molecule per microliter. إذا حطيت بدل 100 molecule per microliter حطيت 1000 molecule, so I increase the concentration, obviously you will kill more bacteria. إذا خليته 100 بس حطيته بدل ساعة حطيته 10 ساعات, you are giving more time for these 100 molecules per 1 microliter, whatever, to kill more cells. So the length of time and the concentration, both of them increase the rate of killing of microorganisms. Chemical agents, هلا بنحكي عن all the agents, بنحكي عنهم one by one. The first one that we are going to cover are the chemical agents. Chemical agents, the mode of action of chemical agents refers to their variable ability to function as killing agents. So if you want to standardize هدول الagents, في عندي كتير أنا مواد كيماوية كتير. There are a huge amount or huge number of chemical substances that can destroy bacteria. So عشان تعمل standardization لهم, we use something called the phenol coefficient. So this is the مقارنة أو ratio of concentration of phenol, who one of the first agents that was used as a chemical agent for this as a disinfectant. بجيب شغلة مثلا the same concentration, بشوف how much killing am I able to kill with the new agent in comparison to phenol. it's just comparing مثلا هذا واحد الفينول, هذا بقتل 100 ضعف عن الفينول, هذا بقتل مليون ضعف عن الفينول, هذا نص الفينول أو ربعه أو 1 على 100 مثلا, وهكذا. So this is a standardization method. So chemical agents act primarily on one of 3 mechanisms that they are able to use to destroy cells. شو في عندي أنا بالسيل؟ DNA, proteins, cell membrane. راح تشوفوا كمان أو cell wall. راح تشوفوا كمان الأنتي ميكروبز كمان بيشتغلوا anti DNA, anti proteins, anti cell wall. It's the same thing. So chemical agents act primarily on these 3 main structures اللي هم the main structures of الخلية, the essential structures تبعين الخلية. So destroy the cell membrane or the cell wall. It is a selective barrier حكينا. Where compounds are actively transported, you have enzymes that are anchored, you have your electron transport chain, you have lots of proteins that are important. So it is the target of lots of substances that can concentrate in the cell membrane and result in killing. طبعا أهم شيء إنه it is containing all the cellular cytosol, كل البروتينز جوا الخلية. إذا كسرته كمان, obviously that will destroy the cell. Osmotic lysis is the main threat اللي هو the main threat of the cell wall function to prevent حكينا. It is maintaining the cell pressure, the osmotic pressure, as well as the حكينا إنه the shape of the bacterial cell is also from the cell wall. So osmotic lysis, the bottom line is they do not lyse the cell. They remove the structure which prevents the cell from lysis due to osmotic pressure. هدول هم اللي بنحكي. شو يعني؟ إنه if I target the cell wall and I remove the cell wall, the cell becomes prone to the osmotic pressures. إذا صار في osmotic pressure كتير مثلا salt or reduced salt راح تصير مثلا it will swell or shrink according to the osmotic pressure حواليها, ف it might be prone to lysis. If you can control by some mechanism, you can control the osmotic pressure and you remove the cell wall, the cell might survive. That's what we're trying to say. So protein denaturation, protein to function properly إنه هم هذا حكينا بالجروث, حكينا إنه لازم يكون مثلا في مثلا حرارة, salt, pH, whatever, all of them what makes a bacteria able to survive اللي هو these mechanisms that maintain the folded 3D structure of their proteins. هاي الخلية بتعيش في 37 because its proteins are folded correctly in 37. على مثلا بترفع درجة حرارة الجسم ل 42, هدول البروتينز they are misfolded and the cell is unable to function and might die. So that's protein denaturation. So you are removing the tertiary structures of the proteins by these agents. فمثلا physical agents like heat, pH, chemical agents like alcohol might denature proteins and cause them to stop working. DNA damage. Now these are agents زي ما حكينا بمحاضرة الجينetics. So you are destroying, you putting DNA mutations or disrupting DNA. حكينا عن ionizing radiation, ultraviolet light, DNA reactive chemicals. راح نعيد هنا كمان شوية. Among the last category are alkylating agents زي ما حكينا, and these compounds are react covalently with bases. مثلا زي ما حكينا البنزبyrene to form DNA cross-linking. حكينا how the polymerase will make mistakes this way. Radiation damage. DNA الذكر حكينا UV light increases cross-linking between adjacent pyrimidines, هدول الدimers, and then you will have all these loops that makes the polymerase makes lots of mistakes. حكينا how that is in skin cancer. And X-rays also produce breaks in the single and double bond strands and cause DNA mutation or DNA damage. Radiation-induced and chemically induced DNA damage kill the cell mainly by interfering by DNA replication. فأنتم مثلا يعني when you bind the DNA ذكرنا حكينا you bind the DNA, the replication material is unable to come to this bound DNA. في إشي ملزق على هالستراند مش قادر أجي, ف I make a mistake, I jump over this mechanism, I بمشي شوية بقطع أو بعمل mistake أو I don't know what to put, I put G, put A, whatever I can put. So that's how the DNA replication will make mistakes, and the DNA repair حكينا will also make mistakes because of these DNA bound agents. So what are chemical agents? Disruption of cell membrane. The first thing that you use for disruption of cell membrane, and one of the most commonly used things, is alcohol. اللي هو الكحول اللي بنستخدمه مثلا rubbing alcohol بسموه, which you use to عادة بيكون مثلا in clear container with a blue مثلا عندنا blue cap عادة بنستخدمه. So this is عادة مستخدم as an antiseptic to clean the skin, and also can be used as a disinfectant. عادة as it is not harmful to the skin. You can clean the skin زي ما حكينا before. Small punctures. The main mechanism has both mechanisms. It can destabilize the hydrophobic structures of the cell membrane as well as denature proteins. It must be mixed with water for optimal activity. راح تشوفوا هذا this is تنعاد هاي this theme بنعاد معنا كتير من هون وطالع, إنه if I have in any of my antiseptic or disinfectants, if I have both hydrophobic and hydrophilic interactions مع بعض, I will be able to destroy the cell membrane. إذا عندي شغلة بس hydrophobic or only hydrophilic, I will not be able to. Why? Because you know the membrane, the lipid bilayer, is hydrophobic on one side and hydrophilic on the other side. So if I can the way I disrupt this, if I have something hydrophobic to bind to the hydrophobic and something hydrophilic to bind the hydrophilic, in this way I can unravel the cell membrane and cause the cell membrane to disrupt the cell membrane. So بدل ما استخدم هل شو اللي راح يكون معناته more effective if I use 100% alcohol ولا 70% alcohol و30% مية. مثلا الكورونا فيروس dies at 76% alcohol مش 100% alcohol. Because of this ability you are able to disrupt this mechanism between the hydrophobic and hydrophilic parts of capsules, cell walls, cell membranes, and all that. So it's not as effective as iodine containing compounds for larger skin punctures. فمعناته ما بتقدر تستخدمه مثلا للاشياء كبيره كاثيتر او بلاد كلتشر او شيء، فبتستخدموا اشياء اللي مثلا عاده سمول نيدل اللي تسحب دم بدك، عاده نسحب عينات دم، هذا اكثر شيء بنستخدمه لتستخدم الكحول for. Ethyl alcohol, isopropyl alcohol are broad spectrum, but they are not sporicidal. هذا مهم. Detergents. شو أنواع detergents that we use? Detergents هم زي ما حكينا that you need hydrophobic and hydrophilic portions on the same molecule. الإيثانول is all hydrophobic، فعشان هيك إذا ضفت معاه مية I will give it the ability to interact with hydrophilic parts. Detergents, the chemical compound itself has portions that are hydrophobic and portions that are hydrophilic. So it is effective also in disrupting cell membranes and makes them obviously lipid soluble. And then similar to other disinfectants, they may harm health in a variety of ways. That's why they are عادة لا يستخدموا detergents على جسم الإنسان. They are used as disinfectants. These surfactants interact with lipid portion of cell membranes and they disrupt the composition of the bilayer زي ما حكينا. أهم شيء في detergents اللي هم يسموه QACs أو quaternary ammonium compounds اللي هم هلا بورجيكم تعرفوهم. So these are the things that are used to clean surfaces, they floors, Lysol, Dettol، أكيد بتعرفوا الديتول. هدول هم ال QACs quaternary ammonium compounds. فهل بيستخدموا على الجلد؟ هل بيستخدموا داخل الجسم؟ Obviously you know that these are not. They are sporostatic and they can use as disinfectants عادة to clean hard surfaces like we mentioned. QACs are mycobacteriostatic as well, sporostatic, but they have an effect on lipid envelope, but not non-lipid envelope viruses. Why? Because of the ability to disrupt they have the hydrophobic and hydrophilic portions. So Dettol, Lysol, whatever you use, these are the عادة the common QACs الموجودين بالسوق. What are the side effects of these QACs or quats? They range from mild skin and respiratory irritation. They can also cause severe burns, can cause nausea, vomiting. In severe cases and higher can cause even convulsions and hypotension and shock. Most commonly found اللي هو بتشوفوا contact dermatitis عادة اللي بتنظف بيتها أو اللي بيشتغل بتنظيف عادة they might have a lot of dermatitis on their hand. It is a common cause من هو من الكيماويات بتستخدمها عادة الكيماويات اللي هم are the QACs. فينول حكينا شو هو الفينول، حكينا الفينول coefficient. Phenol is the very first disinfectant used in an operating room. لما بلشنا نعرف إنه في إشي اسمه bacteria and these can kill الناس when we can't open up bodies and people's bodies without causing an infection. بلشنا يستخدموا الفينولز and this is the very first disinfectant to clean an operating room. Chlorhexidine راح تشوفوا chlorhexidine it is now the most commonly used surgical rub. يعني لما يجي الجراح بده يشتغل أو إذا أنت اشتغلتوا بالجراحة بدك to scrub your hand before using يعني عادة this is for major surgeries, probably not for fast dental surgery, but maybe something like maxillofacial surgery. You will have to use a surgical scrub. The surgical scrub is chlorhexidine. It is a chlorinated phenol. It is widely used as وهذا لونه زي هيك. يعني memorize this. This is what chlorhexidine is. This is the surgical hand wash. Hexachlorophene is a biphenol which with 6 chlorine atoms, but it is used in antimicrobial soaps عادة, but it has been limited now because it has a lot of neurotoxicity. They are lipids in nature and taken up by cells, and they can denature proteins as well. Again, mycobacteria with the lipid-rich waxy envelope are generally highly resistant to these compounds. So you need, because of the high lipid base, يعني has too much lipid that it is more harder to deal with them. It can still kill them, but it is more resistant. ففي شيء اسمه chlorhexidine gluconate which is this is عادة بيستخدموه as a non-alcohol form of mouthwash. راح أتوقع فيه كمان عن لقدام شوية. It is used as a mouthwash. Look at this, it's a small, very small concentration, 0.2% weight for volume only. Whereas this is used about 4% weight for volume, and it is still chlorhexidine gluconate as well. So the bisphenol are very widely used as components in antiseptic hand wash. هذول كمان اللي تسمعوا عنه هذول هم اللي نستخدمهم احنا السوبس نستخدمهم. This is the main thing that is used. Triclosan is one of the things. It is antibacterial, sporostatic, and hexachlorophene as well is used. dental use حكينا ان toxic chlorhexidine كانوا يستخدموا بالروت كانال بالirrigation and intracanal dressing, but now they are using sodium hypochlorite bleach and has been replaced with that. I think this is what you use now in your lab. حكينا chlorhexidine mouthwash with combination with normal tooth care has been shown to reduce the buildup of plaque formation and also prove mild gingivitis. These have a number of side effects including damage to the mouth, lining, tooth discoloration, and tartar buildup. Topical use, they have been used successfully in developing countries. هلا هذا يعني one of the things that I like to show. مثلا استخدموا هذا surgical scrub. هي البيبر تحت. So النيبال واحدة من الأشياء الدول بتقيس فيها مدى الإفكت تبع its healthcare system. How good is your healthcare system? How good are your hospitals and all these things. بتشوف مثلا how long do people live, and one of the things are maternal mortality يعني خلال الولادة and infant mortality. شيء من infant mortality. So دولة نيبال كان عندهم a huge number of infant mortality, and they found that a lot of it was due to an omphalitis, an infection of the umbilical cord. They were able to reduce neonatal mortality, يعني نقلوا حالهم نقلة نوعية بالhealth care effectivity by reducing neonatal mortality by 12%. بإيش عملوا؟ شو عملوا؟ The only thing that they use is that they started using surgical scrub to scrub the babies after cutting the umbilical cord. لأنهم كانوا ما يستخدموا أشياء sterile and clean to cut the umbilical cord. فبما إنهم they were not able to sterilize their equipment, راحوا أعطوا hospitals that deliver babies إنه أعطوهم surgical scrub. أقل ما فيها use a surgical scrub after you have delivered the baby, and that has reduced their neonatal mortality by 12%. So membrane حكينا QAC زائد phenol زائد alcohol. والبروتينز you use chlorine plus iodine. هلا نحكي عن الايودين نجي لقدام شوي. So chlorine, now we are beginning talking about modification of proteins. Chlorine is used as a disinfectant. بتعرفوا الكلورين مش مثلا بيحكوا مثلا هاي المية فيها كلور صح؟ البراك والواتر سبلاي عادة موجود بيستخدم فيه كلورين. Chlorine is a common disinfectant to purify water supply. It is also the same active component بالبليتش كلوروكس المستخدمه اللي هو ما بعرف شو اسمه عندنا بالبلد. بتوقع كلوروكس عندنا بالبلد اللي هو البليتش. فهو ما يستخدم عندنا يعني اللي هو البليتش كلوروكس whatever. It's the same thing that is used for the chlorine. It is just the function is essentially the same. It's just a very powerful oxidizing agent that causes severe cross-linking of enzymes. It is extremely toxic. Obviously, if ingested, it will cause huge damage and huge destruction of cells, beginning from the liver, the esophagus, the stomach, and is obviously very, very horrible. So bleach عادة بصير يحكوا مثلا إنه be careful not to mix bleach with alcohol, bleach with QACs or anything because it can cause harmful compounds that can become that can fly up and can be breathed and cause inhalational injury. نحكي هلا عن next thing we'll be talking about is iodine. Iodine هلا اللي هو is the most common thing that هلا بما ان الكلورهكسيدين هو المستخدم as a surgical scrub to prepare the patient. I don't use chlorhexidine to prepare the patient's skin for the surgery. What I really use is iodine. اتوقع اكيد شفتوا iodine قبل هيك اللي هو هذا يكون لونه احمر. This is what surgeons use to prepare the skin for incision. Iodine is the most effective skin antiseptic used in medical practice, and it is always used prior to obtaining a blood culture. Anything larger than a needle that you want to take from a small needle, bore needle that you use for withdrawing blood or venipuncture, anything larger than that you need to use some form of iodine. iodine هون بنحكي فيه احنا قاعدين نحكي عنه مع ال agents that affect the proteins, so they are oxidants that inactivate sulfide-containing enzymes against the proteins and bind specifically to tyrosine residues in the proteins. However, it actually how it kills is not completely agreed upon. It interferes with the proteins in the electron transport chain and also cause structural disruption for the cell wall proteins as well. Iodine فيه two forms. في tincture اللي هو الweak iodine راح يكون 2% iodine. هذا هو بتستخدم إذا بدك تستخدم blood culture. ما راح تعمل هذا كله in a blood culture. بدك تسحب blood culture أو تحط catheter. بتجيب هذا اللي هو tincture of iodine. It's just 2% iodine. Can be used also for first aid, and it should also be removed with alcohol because it is an irritant. It can cause irritation for the skin. And the other thing is the actual iodine, the iodophores that are used, or PVP, povidone, اللي هو يستخدم for the skin, to prepare the skin for surgery. كتير مهم نحكي عن النقطة اللي لما حكينا إنه the rate of killing requires time and requires concentration. فقد ما تقدر تحط أنت على قطعة الجوز قد ما تقدر تحط عليها ما راح يعني this is the most concentration you're going to put. So what else can you do to control the rate of killing? اللي هو time. So everything that you use من mouthwash, alcohol, chlorhexidine, whatever you use. لما نحكي للناس إحنا مثلا غسل إيديك 40 ثانية وأنت تستخدم soap to reduce the risk of corona, whatever. What are we doing? We cannot increase the concentration. شو بنعمل? We are increasing the time. So everything that you have to give it time, you have to give it at least 20 seconds to work, a minute. مثلا إذا كان هون you want to achieve higher reduction of the cells, so you need to give it at least a minute maybe or two to cause more killing of the skin bacteria. So everything is allowed. حتى mouthwash مثلا إذا مثلا mouthwash شو أحسن تستخدم هيك تحطه بتمك تغسل هيك ثانيتين وتكبه ولا تضلك keep going for 30 seconds. Obviously that more time is more effective. So صار يحكوا مثلا إنه proportion of patients with surgical site infection according to type infection. This is one of the papers that try to compare between chlorhexidine and iodine. And they found مثلا any surgical site infection مثلا chlorhexidine أقل من iodine. Superficial كمان أقل. Deep incision infection أقل. Organ space is the same. Sepsis is برضو أقل. ف maybe using chlorhexidine in some يعني لسه في دراسات بتحكي مثلا maybe using chlorhexidine at the surgical scrub to prepare the patient is might be better. But again, you will look into other papers. You will find papers that support the other. You're maintaining using the iodine. So again, like we said, what are the uses? The most uses are for the pre and post-operative skin cleansing, to prevent infections in wounds, واحد صار معه wound cut، عندهم ulcers diabetic ulcers مثلا burn patients. All these people have lost their main اللي هو skin. They lost the most common or the most important, one of the most important innate mechanisms to prevent a huge influx of bacteria and microorganisms into your system, the whole skin. So one of the things that you can use for these wounds and cuts and burns and ulcers is iodine. They can be used in a variety of concentrations زي ما حكينا من 2 to 10%. And there are solution spray and scrub and ointment. PVPI اللي can be used to prevent neonatal conjunctivitis رح نحكي عنها can be given as a drop in a buffered manner. Both are STDs. إذا كان المother has an STD, an active STD والبيبي طلع من هالكنال راح يتعرض obviously for chlamydia and gonorrhea, which are the sexual transmitted diseases. But they might go through the eye and cause conjunctivitis. One of the things that people we can use is these eye drops. One of them is this PVPI, but راح تشوفوا إنه we use other mechanisms for these as eye drops. نقطة كتير مهمة إلى الآن iodine and to some extent I think also chlorhexidine هدول الأشياء that denature proteins bacteria do not develop resistance to. ليش؟ Because these are general mechanisms that can destroy all proteins بالخلية. So I cannot. There's no way for me to have a mutation that is beneficial against all كل البروتينات تبعتي. يعني زي كأني مثلا ما بقدر أحط بكتيريا بتعيش بال37 أحطها ب100. There's no way لو ضليت من هون لبعد بكرة الصبح أحط بكتيريا واستنى واحدة فيها تعيش ولا واحدة رح تعيش. Why? Because they do not حكينا their proteins do not fold correctly in that temperature. البكتيريا اللي its proteins fold correctly in 100 مثلا درجة حرارة 100 هي راح تعيش بهذيك temperature. So this is the same manner. Iodine, chlorhexidine, they cause a widespread destruction of proteins, and there's no amount of mutation that may change the bacterial structure or makeup of all their proteins. إلى الآن ما فيش resistance for the iodine, probably زي ما حكينا because it probably targets multiple pathways and multiple proteins all at the same time. هذا هو iodine. Iodine acts very quickly. It's not instant زي ما حكينا. It still needs time. نشتغل. Has a major advantage that it makes it ideal for surgery because it is bactericidal, fungicidal, tuberculosidal, virucidal, and in reports بنحكي كمان is even sporicidal, can even destroy some spores. Iodophores are complexes. هلا شو الاسم رح تسمع عنه بالمستشفى بسموه بيتادين. عندنا وكثير من المستشفيات ودول بيتادين. هذا مش فرقوه عن بيتادين هذا it's a morphine derivative. فهذا بيتادين اللي هو يستخدم اللي هو الايودين. فهاي مثلا واحدة من الدراسات بيحكي لك effect of povidone and chlorhexidine mouthwash on plaques for Streptococcus mutans مثلا. فوجدوا إنه مثلا يعني look at the main thing in the results is that you are trying to establish a favorable environment بالmouth to reduce plaque formation and healthy flora. So من تتذكر إنه fixing flora to be healthy is better than eliminating. تذكر حكينا I don't want to remove all bacteria because other bacteria will always come in its place. I want to balance bacteria. So وجدوا إنه مثلا result إنه مثلا هنا immediately after mouth rinse therapy وجدوا إنه مثلا reduction بال Streptococcus mutans, which is the main thing that can cause these plaques. And they did the same thing the iodine rinse, chlorhexidine rinse, or placebo. But they found that gradually after 3 months, bacterial counts in the PVP group were the same as post-operative counts. So conclusion, in مثلا chlorhexidine can be recommended as it is shown to have better effect than as a mouthwash. So this is just one of the studies that was shown here. فهذا this is one of the common bacteria found in the oral and oropharyngeal pathogens, and just to show that I need to control all these. هدول طبعا من just يعني رؤوس أقلام. There's a lot more than this. Comparison. Another comparison. طبعا هدول ما حد يسألني هدول داخلين امتحانات. This is a comparison just to show a just to show يعني to showcase المعلومة and just to broaden الفكرة شوية. So maintenance as an antiseptic for maintenance of oral health as well. This is something very similar to the previous paper as well. The same thing here. Fungicidal activity, virucidal activity, all of it is just destruction of other benefits that you can use iodine for. It has been shown as to reduce spontaneous bleeding following tooth extraction, significant reduction of swelling after molar removal, reduced oral caries in susceptible children as a mouthwash, antiseptic for these special cones that are used in practice, and it does not discolor the teeth زي ما بنعمل ممكن يكون بالchlorhexidine as well. Let's stop here. We'll continue the next lecture inshallah from here, and we will see you then inshallah.